Good evening to all my wonderful champions. Actually, not exactly good evening, good afternoon. For it to be evening, we need one more hour, but yeah. Hello, children. So this is going to be the first live regarding what do you call the theoretical aspect or the study aspect, my dear children. And I'm really excited to see that. How many of you are gonna join? So I see 3 people already up there. Okay, so if at all you are someone who is watching me, I'll request you to please share the live link to your friends also who have opted for biology. Hi Asia. Good afternoon, dear. Okay, so we'll just wait for some time so that everyone else can also join. If at all there is at least some people who have taken biology, at least let them join, okay? Let's wait for that. I hope you're good. Everyone out there. Hello, Hidden Stories. Hello, dear child. So my dear children, I am pretty sure that you might be a bit stressed up because of your exams. Okay, so one thing to say, whatever has to come, right? Whatever will happen, will happen. So definitely do not forget to give your best in all those situations. Okay, so my parents, okay, my father used to tell me, Jasmine, you just have 5 seconds to take a decision. Whether it can be a yes or it can be a no. So take this decision of giving your 100% efforts into studies, my dear children, as of now, and be with me for the whole live video. I am going to make sure that you get really good marks in your unit test. Okay? So I hope you are ready. I see few people have joined, so let's begin. Yeah, before beginning up, one very important thing, whoever have not clicked on the subscribe button, click on it right away. Okay, we need more of you to participate in this whole process. Hi Spurti, big hi. Chalo, my dear children, so I am going to start off with one of the most important chapter out there. As I promised, I will be starting with principles of inheritance and variation. Now, why is this chapter important? Because it gives you 12 marks or 13 marks in your PU2, which is a big number, and also for your KCT examination. But as of now, let's focus on PU2. Okay, so are we ready to begin this live session? Can we go ahead? Can we begin this live session? If at all we are ready, just give me a thumbs up. Your channel is really helped me. Today is my chemistry exam. I have done really well. So all thanks to Ashwarya ma'am. Thank you so much, Asya, for coming and telling that over here. So my dear children, who all are ready, give me a thumbs up and we'll get. Okay, Spurti has given me a thumbs up. So this should be not a one-way lecture, okay, a two-way lecture. It should be a two-way lecture. We both should be interacting with each other. So since one person had given me thumbs up, I will be moving ahead with the first question out there, and that is this. I hope it's clear. In a monohybrid cross between 2 heterozygous individuals, my dear children, okay, percentage of pure homozygous individuals obtained in F1 generation will be what? Asya also has given thumbs up. In a monohybrid cross between 2 heterozygous individuals, heterozygous means the gametes are not the same. Okay, for example, if you have a condition that is something like this, capital T, small t, this is heterozygous condition. Is this visible? Children, are you able to see this? The screen? Is the screen visible to you? Come on. Answer is B. Spurti said the answer is B. Okay, let's see. Anyways, let me decode since you have not telling me whether you're able to see or not. So, 2 heterozygous individuals, percentage of pure homozygous individuals obtained in F1 generation. So, heterozygous means, for example, I take capital T and small t. If I am not visible, just give me a second. I'll be right back. Just a second, okay? Okay, now I think it's pretty decent. Hello Shri Girish. So over here we have taken 2 heterozygous individual, capital T, small t, and again capital T, small t, and then we have to cross it. So these are the parents. Now the gametes, it would be capital T over here, it can be small t. Similarly here capital T and small t. We cross them. Suppose this gamete fuses with this, I get capital T, capital T. This fuses with this, I get capital T, small t. This fuses with this, I get capital T, small t. And this fuses with this, I get small t, small t. So you have been asked one very important question, that is, how many homozygous individuals will be obtained? By homozygous means the gamete, the over here, this particular thing, the alleles which are there, it should be same. So over here I see capital T same. Here also I see small t, homozygous condition is there. So out of 4, 2 of them are homozygous. Out of 4, half of them, 2 means half of 4 is 2, right? So half of them, that means 50% of them are homozygous. So your answer would be B. And let me see who all got your answers correct. Sri Girish said B. Spurti, for the first time itself, she said B. Kudos to you, Spurti. So we have to climb up the leaderboard, okay? Going ahead, I hope this question is clear to you. From this point onwards, we are going to be super energetic. Now, question number 2. What is the possible blood group of an offspring when both parents have AB blood group? When both the parents have AB blood group, my dear children. So, parent 1, my dear children, is AB. Parent 2 is also AB. In that case, the gametes that it will be producing is what? A and B. Here also this parent A and B. That means half of the gametes can have A gene, half of the gametes can have V gene. Now suppose this sperm and this egg came together, so it will be AA. Suppose this and this came together, then it will be AB. This and this came together, again AB. This and this came together, we get BB. Now we have to tell what is a blood group. So all of you are telling the answers. Purti and Girish said B, but I have to check it. So let's see. So what is the possible blood group of offspring when both parents have AB blood group? So I see it can be A blood group because it is capital A, capital A. Here it can be AB blood group, or it can be B blood group also. So in that case, the answer would be A, B and AB. Both of you, Spurti and Sri Girish, kudos to you. Kudos. The answer is what? Question number 2's answer is B. So superb out there. Now other children who are watching this video and not participating, it's high time to participate. Either you give me a wrong answer or the correct answer, but the solution to it, whatever you get, my dear children, you learn from it, right? So please participate. Now in the F2 generation of Mendelian dihybrid cross, the number of phenotypes and genotypes are the number of phenotypes. When Mendel did the dihybrid cross experiment, okay, and when he went ahead with the second F2 generation, my dear children, he got certain things. He tried to cross round and yellow plants with wrinkled and green plants. Okay, now the question says, what is the number of phenotypes? What is a new physical character that you saw over there? Or what is a general physical character that you saw in the dihybrid cross? So first general character is I saw round and yellow plants, and I also saw wrinkled and green. Then I got something else. I found round and green, that is a new combination. And also I found wrinkled and yellow. So how many phenotypes, how many physical characters? 1, 2, 3 and 4. If you are asked about the ratio, how will you tell the phenotypic ratio? 9 of them were the dominant trait, that is the round and yellow one. 1 of them were the recessive character, that is wrinkled and green. And you got 3 new combination. 3 new combination. So the phenotypic ratio is 9:3:3:1, my dear children. Now you are asked, how many genotypes were there? So I see Preeti also have come into the picture, okay, and Spurti and Girish have been giving me 2 answers. Let's see whose answer is correct. So Spurti says the answer is A. Okay, Spurti says the answer is A. Sri Girish says the answer is B. What if I tell you both the answers are wrong? My dear children, in F2 generation, you saw 4 phenotypes for sure. 1, 2, 3, 4. So wherever 4 you see, that would be correct. This option would be anyway wrong. B option is anyway wrong. Now if you talk about the genotypes, actually you have just 9 genotypes. You just have 9 genotypes, genetic makeup. For example, it can be capital A, capital A, capital Y, capital Y. Tell you that this is round yellow. Now if this is the case, then also it is round and yellow. If this is the case, then also it is round and yellow. Now these genotypes, my dear children, were repeated but you are not asked about the repeated numbers. You are asked what were the unique genotypes that you saw. So this one, this two, this three, and so many. How many of them? 9 of them. 9 of them. So your answer is D. Your answer is D. And since I know it's difficult to remember in that sense, I have put it down over here. These were the unique genotypes. Capital R, capital R, capital Y, capital Y, capital R, capital R, capital Y, small y, and all the others. 9 of them were there, 9 genotypes. Okay? Oh no. Yes. So your answer would be D. Yes, Preeti, it's D. So it's totally okay to get it wrong. As I told you, you can get it wrong now, but not on the day of examination, that is tomorrow, right? So please give your best. You are going to be on the top. Fine. Now, my dear children, if you're asked, in total, how many plants did he get? So the answer would be 16. In total, he got 16 plants. 9 of them, I told you, they were round and yellow. 1 of them, they were wrinkled and green. 3 of them, they were round and green. Rather than being round and yellow, round and green. Over here, rather than being wrinkled and green, they were wrinkled and yellow. So 9 plus 3 plus 3 plus 1 is what? 16 progeny they got. Okay, they got 16 progeny. Hello, Ganesh. So your answer is D. Now going ahead with the 4th question. Let's see who gets this particular question correct. Okay. 37.2% recombinant Drosophila progeny obtained in the T.H. Morgan's dihybrid cross experiment with the phenotypes red eye color, normal body, and white eye color, miniature body is due to. So they are asking you a question, that is, we see 37.2% recombination. How do you get this much recombination? That is the question. Now, they have twisted it with a lot of other data out there, and I know it's too difficult for you to understand. I'll tell you one very, very simple thing. That is, suppose, okay, you have a best friend. You have a best friend and you guys are really close. Do you think some other person can get over here and do the third wheeling thing? No, they cannot. So similarly, over here, my dear children, suppose this is a chromosome and this is a chromosome. This is you and your best friend. Okay, this is you and your best friend, and you are really, really close to each other. Now, whatever you do, my dear children, wherever you go, suppose there is a condition where the chromosome exchange their material. But in that case also, what will happen? Suppose this part of the chromosomes are being exchanged. These guys will be still together. These genes will be still together, right? Because they are very close to each other. So I'll call them linked genes. I'll call them linked genes. Do I see any new combination? No, ma'am, because these guys are always together. Suppose you and your so-called best friend are actually not that close. Okay, now what will happen? Suppose the parts have to be exchanged. In that particular condition, my dear children, it can be something like this. This part can go over here. This part can remain over here itself. Now, do I get a new combination? The answer is yes. So, in this case, I say the distance would be more. When the distance is more, I will be able to find more new combination or more recombination. So when I say 37.2%, ma'am, is it too much recombination or less recombination? The answer is too much recombination. 50% is the highest. Now if something is showing 37.2%, that means these genes are not linked together very tightly. They are loosely linked. What do you say? They are loosely linked. In this condition, we say that they are tightly linked. In this condition, we say that they are loosely linked. And loosely linked genes would be far away from each other. There will be too much distance. So over here, if they are showing this much recombination, then they have to be loosely linked. So I will cut all the options with tightly linked. Okay. Now loosely linked and shorter distance between the genes. Can something be loosely linked and can have less distance? The answer is wrong. It has to be loosely linked and what? Longer distance between the genes. So your answer would be C, my dear children. Okay. Now if in the question, yes, Spurti, the answer is C. So Spurti has been saying C for a long time. Asya, one more try. Okay. We can do once more. So I know some people know, some children, they read just one part of the question and they go ahead with answering, but then this is a learning for you. We have to read the whole option. Suppose if in the question the thing was 1.7% recombination. If something was showing 1.7% recombination, in that condition, what would have been your answer? Now this is homework. Right now I want the answer for this homework. Okay. Suppose in the question it was given 1.7% recombination is there. Then in that condition, what would be your answer from here itself, from the choices given over here? B. B. Tightly linked and shorter distance between the genes. Exactly. So you have got the concept. Clear. Okay, this is the concept and it's easy. Remember it by your best friend. Okay. Fine. Now going ahead with the next question, my dear children. Fifth question. In a dihybrid cross in pea plants, Mendel got 9:3:3:1 phenotypic ratio. It denotes that. It denotes what? What does it denote? That the alleles of two genes are interacting with each other. Is it? Is it like that? My dear children, I told you. What does 9:3:3:1 mean? First thing, 9 means 9 of them are round yellow. I'm writing it again because I want to explain something. 1 means you're talking about the recessive trait, that is wrinkled and green. Now over here you have to add the new things. Okay, so round was with yellow, so we have to make round with green now. That is 3. Here wrinkled was with green, so this time we would make it with yellow. Wrinkled and yellow, right? Now over here, my dear children, so Asya has given the answer D. Spurti also gave the answer D. Okay, anyways, let me explain to those children who do not understand. Now, when you are saying you get new combination, that means you understand one very important thing, that all the alleles are independently getting segregated. They are all getting segregated independently. Otherwise, in the progeny generation, you would have only seen round and yellow. But that's not the case. You are seeing new combination, my dear children. So the alleles of two genes are interacting with each other. The answer is no, they are not interacting. It is polygenic inheritance. The answer is no. Polygenic means you need, for one particular character, you need many genes. No, it's not like that. It is a multiple allelic inheritance. Do you have so many alleles? No. The alleles of two genes are segregating independently. This is absolutely correct. Round and green has nothing to do with each other. Round can go with yellow, round can go with green also. Okay, so your answer would be D. So those of you who got answered as D, that is Purti and Asya, kudos to you. You are doing a wonderful job out there. Now, my dear children, as I told you, as of now, we have just started off the session. There can be so many people who can benefit from the session. So, if at all you have your friends around, please send the link to them. Ask them to join, okay, and watch this video. Now, going ahead with the 6th question, we are ready, I suppose. Mendel's hybridization experiment material was very easy. Chalo, everyone answer. Come on, 6th question. Spurti says the answer is A. Okay, let's see what Ganesh, what Sri Girish, what's Preeti and Asi has to say. Other children, my dear children, on what thing did Mendel do his experiment? We know it's garden pea, right? What's the other name of garden pea? Any idea? Any idea? What's the other name of garden pea? The scientific name of garden pea is Pisum sativum. So the answer would be A. It would be A. Pisum sativum. Somashekar, yes. Preeti, yes. Now I see people are coming and answering, and that's super awesome. It is Pisum sativum, my dear children. Please do not forget it. Okay, it's a very easy question. Now going ahead with the next question. Let's try this one. Okay, all of you. Question number 7. In 19,090 AD, 3 biologists independently discovered what? Okay, discovered what? In 19,090 AD, 3 biologists independently discovered what? Discovered, sorry, not what. 3 biologists independently discovered Mendel's experiment. Okay, so who were those scientists? That was the question. So is it D. Rice, Correns and Schemark? Is it study of one character at one time? Is it Avery, MacLeod and McCarthy? Is it Bateson, Punnett and Bridges? Asya, Preeti, Somashekar, Spurti, everyone says A for this question. Children, can you also put the question number? It will be easy for me to understand. So I believe you're saying this, the answer is this itself. It was D. Rice, Correns and Schemark. You know this question is also very, very important. And from which year to which year did, Mendel did his experiment? What is the answer for that? From which year to which year did Mendel do his experiment? So it was from 1856 to 1863, my dear children. For 7 years he did his experiments. Please remember the year. Now post that, after so many years, came 3 biologists who reworked on Mendel's experiments. Okay, so it was D. Rice, Correns and Schemark. It's given in your NCERT. Go through it. So 7, A, Preeti, A, Asya, A, good. Kudos to all my children out there. So I'm seeing girl power over here. What about the boys out there? Chalo, let's solve this. What type of gametes will be formed by the genotype capital R, small r, capital Y, small y? What type of gametes would be formed? How do you find the gametes, my dear children? Always remember, when something is given like this, Okay, you have to use your imagination. That means you have chromosome, one with capital R, the other one with small r. And then you have a chromosome, one with capital Y and the other one with the small y. Now, whenever we have studied option, Nagaraj, okay, so one boy has come over here, Sonu, okay. So I see boy power also now. Spurti says 8A, let's see if it's A. Asya says A, okay. So my dear children, imagine these type of chromosomes. Now, whenever the chromosomes, they separate and they go into gametes, from one pair, one guy can come in. So capital R can go with capital Y, agreed? Capital R can also go with small y. Similarly, small r can go with capital Y and small r can go with small y. So your answer for this question would be A. So who all got it correct? Let me see that. It was Nagaraj, it was Asya, Spurti, Sonu, all of you got it correct. Kudos to you, my children. Now going ahead with the next question, question number 9. If two with AB blood group marry and have sufficiently large number of children, these children could be classified as A blood group, AB blood group and B blood group in ratio 1:2:1. That means two people who are having AB blood group, they marry. You will be finding that one can have A blood group, one can have B blood group, and two of them, there are chances that they can have what? AB blood group. That is the proportionality. Now, modern technique of protein electrophoresis reveals presence of both A and B proteins in AB blood group individuals. This is an example of what? So Harshini, hi dear, Harshini has given the answer as B. Okay, let's see. Asya says it's A. Spurti says it's A. So, what would be the answer for this? So, it is said that blood group which is there, AB blood group, over here the protein for A and the protein for B, both of them are equally present. That means, what do you say? They are equally dominant. So, we will say that they are co-dominant, and this process is known as co-dominance. A is your answer for this question. Yes, Nagraj. Okay, your answer would be A. Harshini, I am so happy that you tried, and I am pretty sure you will get your answer correct on the day of examination as well. Okay, now going ahead, I hope this is clear, my dear children. So, AB blood group show what? Co-dominance. Very, very important concept. Now going ahead, the frequency of recombination, very important question. The frequency of recombination between gene pairs on the same chromosome as a measure of distance between the genes was explained by. Ma'am, I didn't understand the question itself. So basically, if I told you that the genes which are A and A, they are showing 37% recombination. Okay, I am telling that. But someone else said that if they are showing 37% recombination, then probably that distance would be X. So who was this person who said the frequency of recombination between the two genes pairs on the same chromosome can help in the measure of the distance between the genes? Who said that? Okay, so I see Nagaraj saying option B, Spurti saying, okay, come on, 10th answer, 10th. Asya says it's B, Spurti says it's also B, everyone says it's B. So then, what's stopping me from saying it's B? The answer is B. It is Alfred Sturtevant. He was a student of T.H. Morgan. So children usually get confused between this and this. Remember, the answer for this question is Alfred Sturtevant, because he was the person who introduced the term gene mapping, who spoke about the distance and stuff like that. That if this is the recombination, this much distance would be there. But T.H. Morgan was the person who described about linkage and recombination. He described about that, he taught his student, and then his student came up with gene mapping. So please do not get confused between B and D. Your answer would be B, because here you are specifically talking about gene mapping. Okay. Now 11th question. Let me see who gets this particular question correct. It's not as easy because it's not there in your NCERT. That's all. Otherwise the question is easy. How many types of gametes will be produced by individuals of capital A, capital B, small b and small c genotype? So this is a genotype of an individual. You are being asked this question that how many gametes are going to be produced, my dear children. How many gametes are going to be produced? My dear children, I want all 11 of you to answer this. There are some children who are just watching. They're like, I'll study. No, we do not want that attitude, right? Okay, we have Dilshada Hanchinal. Okay, hi Dilshada. And over here I see the answer is 11B. So you are saying the answer is B for this particular question. Let's check. Asya says it's A. I'll wait for some time so that all of you can answer. This time now I'm not going ahead without getting your answers. Spurti says no, it's not A. Okay. Spurti says it's B. Okay. What about the others? Other 8 children out there? My dear children, this is how you learn. Try to give the answer. Harshini says it's C. Okay. Fine. So I'll tell the answer. I'll tell you how to find the answer. Okay. Or you tell me how to find the answer. I'll give you another question out there so that I'll understand that you understood the concept. Over here, suppose this is a genotype of an organism, capital A, small a, capital B, small b, and small c, small c. I say that the number of gametes of this particular organism, the types of gametes, I can find with the formula, that is 2 raised to the power n. What is n over here? The number of heterozygotes. The number of heterozygotes. So I say capital A and small a is not the same, so they are heterozygotes. So I got one heterozygote. Capital B and small b, they are also not heterozygotes. So this is a second heterozygote. So I've got 2 heterozygotes. This is homozygous. They are what? Homozygous. Small c, small c. So I have got 2 of them. So I'll replace this n by 2. So 2 raised to the power 2, what would be your answer? Your answer would be 4. So in this condition, you can get 4 different type of gametes, my dear children. You have to use this formula. Now, over here, that is not the condition. Over here, this is a condition. You can tell me the answer now. With this formula, try to put in this formula and tell me the answer now. What is the answer for this question? Question number 11th. How many types of gametes will be produced? Chalo, chalo, chalo. Fast. Over here, I have 2 heterozygotes, so I replaced n by 2. If I had 3 of them, I'd replace it by 3. Over here, how many heterozygotes are there? I have capital A, capital B, small b, and small c, small c. How many heterozygotes are there? Abba. Children, try Madi. Spurti says it's A, Harshini says it's D, Nagraj says it's D. Okay, is this a homozygote or heterozygote? Both of them are capital A, so it is a homozygote. Can I consider it? No. Is this a homozygote or heterozygote? Homozygote again, small c. Can I consider it? No. What about them? They are the only heterozygotes. So your answer is, over here is what? 1. The number of heterozygotes is just 1. Now if I put it in the formula 2 raised to the power n, where n is what? Is equals to 1, my dear children. So your answer 2 raised to the power 1 is what? 2 itself. So your answer would be 2. Got it? Did you get it now? You don't have to count like this 1, 2, 3, 4. No. Don't do like that. Just find the pair. How many pair of heterogametes you have? That's all. So 1 pair is there. So your answer would be 2. I hope you got it. If there was a person who gave the wrong answer, but now you have got the concept clearly, give me a thumbs up right now or put hearts over here. Okay, how will I understand whether you are understanding what I'm teaching or not? Okay, can you give me hearts or give me a thumbs up if you understood this? Should I give you one more question like this? Shall I? Okay, I'm getting hearts, so that means you're understanding. I'll give you one more question related to this towards the end. Okay, towards the end of this particular chapter, I'll give that. Now, thank you Spurti, thank you everyone out there. Now, let's see the next question. Recognize the figure and find out the correct matching. Okay, what is this and what is this? Any guesses? This is the 12th question. You have to answer me 12 A, B, C, D. What is the answer? I see multiple. Okay, smiles, scary face, everything is there. Heart is also coming. Kudos. Answer is A. Spurti says A. Asya says yes. Okay. 12th, Nagraj says it's A. Yes, my dear children. Sonu also says it's A. Yes, you're absolutely correct. So in the case of Drosophila, the small one is the male Drosophila and the bigger one is the female Drosophila. So you know what mistake I did? I went and just saw male and female. Okay, and I went with this option. But later I realized it's butterfly that they're talking about. So it's kind of, what do you call, a very easy question, but still we can get confused. So I'm just telling you. So your answer is A, and all of you who gave the correct answer, kudos to you, my lovely children. Now, give me this answer, okay? Question number 13. Which of the following are correct regarding linked genes? Which of them are correct regarding what? Linked genes. So whenever you get a question like correct, incorrect, what do you have to do? You have to underline. what you're searching for. You're looking for the correct statement. So linked genes are the one. Linked genes means the genes which are very close to each other, right? So are located near each other on the same chromosome. Is it correct? Yes, ma'am. They violate the law of independent assortment. What did this law of independent assortment say? That basically if I have two character or genes for two character, both of them will go separately, independently. They are not dependent on each other. But over here, since these genes are linked to each other, can they show independent assortment, my dear children? No, they actually violate independent assortment. So this is also correct. Okay, both of them are together, they do not show independent assortment. Now segregate together during meiosis. So do they move together during meiosis? Meiosis is reductional division, no? From a pair of chromosomes, only one chromosome can move to a gamete. So over here, this chromosome carrying the two genes, they come together, right? Yes. So segregate together during meiosis, that is also correct. So your answer would be D, ma'am. Asya is saying D, ma'am. So yes, the answer is D. Spurti also says D, and kudos to both of you. So my dear children, understand very important thing that you can get questions from all these topics. Now, since I saw I have a less confidence, and also Nagaraj has said that the answer is D. I have less confidence for the other type of question that I gave. So I will give you one more type of question which is out there. Okay, so the genotype of the parent is how many gametes? How many gametes? You can just tell me the answer. Type down the answer and you can tell me. How many gametes would be there if the genotype of the parent plant is this? 8. Okay, Spurti says 8, Harshini says 8. Anyone else want to participate? Come on children, all the 11 children out there who came over here. Nagaraj says 8. I'm so happy. Let's see if it's correct or not. So you have to find the number of heterozygote. This is 1, this is 2. They are also heterozygotes. So 3 of them. Since all of them have different type of gametes, right? Now my dear children, 2 raised to the power n, where n is 3. So in that case, 2 raised to the power 3 is what? 8. So all of you who got 8 as the correct answer. Sonu, top 5 universal. Asya, super great. Okay, doing great. Now my dear children, one more question. Okay, one more question to all of you. Since you have your portions until polygeny and pleiotropy, tell me the correct answer for this. Pleiotropy is shown by, or basically, in which of these conditions do you find pleiotropy to be active? Okay, phenylketonuria, okay, and then hemophilia, and then what do you call sickle cell anemia. Okay, we can remove sickle cell anemia over here. Okay, color blindness, none of the above. None of the above. What would be your answer for this question? This is pleiotropy. When do you see pleiotropy? In which condition do you see pleiotropy? Phenylketonuria, hemophilia, color blindness, none of the above. What is the answer? C5. So C5 is there. So you are saying phenylketonuria. Nagaraj is also saying phenylketonuria, top 5 universal. Hi Prajwal. Top 5 universal is also saying phenylketonuria. Well, let's check if it's phenylketonuria or not. My dear children, listen to me carefully. What is pleiotropy? Pleiotropy is a condition where one gene is responsible for multiple character. Usually we studied capital R is just for round. We didn't study it for, it is for something else, right? But over here, one gene is for multiple character, and the only example is which is given in the NCERT according to this is phenylketonuria, where what happens exactly is that there is an enzyme, okay, whose name is phenylalanine hydroxylase. Phenylalanine hydroxylase, which converts what to what? It converts a amino acid phenylalanine. What is amino acid? Amino acid is something that comes together to form proteins. So there is phenylalanine which is getting converted to another amino acid that is known as tyrosine. With the help of what? With the help of this particular enzyme. Now the sad news is that there is a gene, X gene, which is responsible for the production of this particular enzyme. And this X gene, something happened to it, so it is not working. So do you think this conversion can happen? No, conversion would not be happening. And in that case, this guy would be accumulated. Phenylalanine will be accumulated as phenyl pyruvic acid. And accumulation of this causes one very important thing, that is mental retardation. Mental retardation. So ma'am, you said 2 character, right? Multiple character. Only one character is being hampered. Mental retardation is causing. Is there something else that we need to know? The answer for this question is yes. What is that something? So the second character that is getting hampered because of a mutation in this particular gene is that the tyrosine which is there, it is responsible for the production of melanin. And you know what melanin does? It is responsible for your skin pigmentation. So, since tyrosine is also not there, melanin would not be there, which is why in this condition, the second problem that you would be seeing is what? Hypopigmentation. There will be less pigmentation. Reduced pigmentation in what? In your skin and your hair. Your skin and hair. Okay, got it? So this is one very important question. Please remember, pleiotropy, one gene is equals to multiple character. Now, Harshini, ma'am, 2 marker, 3 marker, 5 marker. Harshini, there is a separate video that I have did on the complete revision where I have discussed all of it. Over here, I'm just for MCQs. Why? Because at this particular point of time, I'll try my maximum to explain you concepts with the help of MCQs, so that at least you can see something. Okay? Fine. Spurti says, ma'am, PS, ECD or MSRIT? Well, that's a tough question. I'll say MSRIT, but I do not know for what reasons are you looking for the college, as in there are so many conditions which applies, right? So I'm not the correct person to say, it's you. Now, my dear children, okay, over here. Chalo, one more question. In which condition do you think that the lightest skin color will be there? Lightest skin color. In which condition? Tell me the answer for this question, okay? Capital A. Okay. B is capital A. C is. D is. Okay. In which condition do you see lightest skin color is there? Now this is not related to your pleiotropy. Okay, I told you in pleiotropy one enzyme is getting hampered, which is why 2 effects are being seen. One is mental retardation, other one is reduced pigmentation of your skin and your hair. But in general, if you tell, my dear children, there is a term that is known as polygyny. What do you mean by polygyny? Multiple genes which are there on chromosomes, my dear children, they are all responsible for one particular trait. So the ulta, the opposite of pleiotropy. There one gene was there coding for multiple traits. Here multiple genes are there coding for one trait. So what would be your answer for this question? I see, yes, Sonu is asking, will the other video be sufficient? Yes, it would be. Just that I have not explained much questions about polygyny and pleiotropy. You can see from here. Okay. So, small, okay, Muskan is also here. I see lots of new people out there. So, yes, my dear children, your answer is correct. Now, for those of you who might be thinking, what does ma'am mean by this particular question? So, for those of you, listen to me carefully. Okay. So, over here, suppose it is capital A, capital A. Capital B, capital B, capital E, C, capital C. In this condition, my dear children, all of the genes will be expressing themselves, and this will be leading to more pigmentation, hyperpigmentation of your skin. So that means darker skin tone will be there. Now suppose here you do not have capital A, instead you have small a. So in that case, your genotype is what? Capital A, small a, capital B, and capital C, capital C. Now this will be lighter than the previous one where it was capital A, capital B, capital C, capital C. Now the lightest skin tone will be there when all of them are recessive. A, B, C, C. All of them small. Okay? I hope this is clear to you now. So your answer would be C. And let me see who all got it correct. So I have got Nagaraj who got it correct. I have got Dina who says option C is correct. Kudos to you. Top 5 universal correct. Asia correct. Muskan correct. So no, thank you so much. And ma'am, I want 35 marks. Then what should I read? You just follow this. I am pretty sure you'll get whole and all. Okay. So listen, first of all, read your NCERT. That's one very important thing. Read every aspect of your NCERT, not leave even a single question. But the thing is, do not be stuck at some place forever. You know, time is very less. You have to utilize that time efficiently. How can you utilize that? By reading a particular thing and solving questions related to it. That is how you can utilize your time. If you get stuck somewhere, you will be stuck there forever. So don't do that. Okay, that's the only suggestion that I want to give. My videos are gonna really help you well. If you can watch the complete revision video, it will be helpful for you. Apart from this is also there. If you have any questions, children, related to this chapter, that is what do you call principles of inheritance and variation, and you want me to explain it, definitely let me know. Okay, before going ahead with the next chapter, one very important thing, I'll tell you the topics that you need to revise. Okay, one is incomplete dominance. Okay, please look for the example of snapdragon, antirhinus, we also call that antirhinus. Please know the genotypic and phenotypic ratio of it. Okay, and then the examples of multiple allelism, that is your blood group and things like that. Okay, and then you have pleiotropy, polygeny. I just explained you. Please go through it once more. Please know the definition of it. Now, chromosomal theory of inheritance, who gave it? It was Sutton and Boveri who gave chromosomal theory of inheritance. What do they say in chromosomal theory of inheritance, my dear children? That basically, whenever you say segregation of chromosome is happening, along with that the genes are also moving together, because where are the genes? They are on the chromosome itself. So wherever the chromosomes will be going, on it are the genes, they will also be moving along with it. Okay, now in which year Gregor Mendel did his experiment? I told you it was from 1856 to what? 1863. For 7 long years he did the experiment. One very important question to you, rapid question, listen to me carefully. Okay, that is, how many characters did Mendel study with his experiments? How many characters did he study with his experiments? Can anyone say? How many characters, children? Come on, come on. Saraswati, I will do everything as you. Okay. 7. Hanumantaraya say 7. Nagaraj say 7. Yes, there were 7 characters. Okay, and is there someone who can tell me the name of all the character? All the character that he studied? Very, very important question, children. You should be knowing all this thing. 7 characters were there. 7 characters were there. If you are asked how many traits are there, 14 traits are there. For each character, he had 2 traits. For example, height. Height means tall and dwarf. That means 2 of them. So 7 into 2. How many traits are there? 14 traits were there. Okay, you should know which one is dominant, which one is recessive, which is the character that he studied, which is the character that he didn't study. All of these things are really, really important. Okay, so if you talk about the color, in color, I think there was flower color. Okay, flower color which was there, violet is there, and then white is there. This is dominant and this is recessive. Okay, if you talk about pod color, there was green and there is yellow. Yellow is recessive. So whenever you look for pea, you look for the green peas, no? When you look for the pod, you look for the green ones. That is what you select. But now when you talk about the seed color, what about it? Seed color, the dominant one is actually yellow and the recessive one is actually green. This is ulta. Okay, this is ulta. Please remember it. Apart from that, you have pod shape. If we talk about the shape out there, so we have 3 of them which are covered. Pod shape. So one is when the pod is full, as in it has all the bees and it's looking like full, complete full. Now one is where the pod is constricted. Okay, the pod looks like this. It's like dry thing. Okay, apart from that, what character, my dear children? Pod shape is done, pod color is done. Now flower position is there. Flower position is there. The dominant one is axial. The recessive one is what? Terminal position. Axial as in, for example, I have a plant over here. From the place where the leaves are coming, there you get the flowers. This is axial position and this is dominant. And terminal means, for example, here I get a branch, from here I get a flower, or from here I get a flower. This is known as terminal position. The place where the leaves and everything comes off, from there if you are getting flowers, my dear children, that is axial flowers. So over here I have got seed shape, seed color. Okay, seed shape. Okay, seed shape is left. Come on, let's find that also. Seed shape. Seed shape is what, my dear children? Over here, round is the one that is dominant and wrinkled is the one that is recessive. So 1, 2, 3, 4, 5, 6. If you see, if we talk about flower only, there is flower color and the flower position. If you talk about pod, pod color and pod shape. Seed color and seed shape. Now one more character, that is your height. Okay, stem height, that is tall and dwarf. Please remember these 7 very, very important things, and especially you can get confused in these 3 things. I'm pretty sure for that. So pod, always remember when you go for shopping, you look for green pods, that is what you take in. Now when you open, you always get green seeds also, but that is not dominant. Yellow is the one that is dominant. Okay, so I see Sonu and Hanuman, Muskan giving me full answers out there. Kudos to all of you and all the other children who have participated. Hanumantreya, Nagaraj, Asya, Saraswati Yadav, Muskan Mehra, all of you doing great, my dear children. So these are the few things that you need to know. Please go ahead, read about linkage and recombination from your NCERT. I have explained it over here. Now going ahead with the next chapter. Okay, shall we go ahead with the next chapter? Give me a thumbs up, my dear children. Are we ready for the next chapter? Okay, no cheating. Listen to me, listen the question and tell me the answer. Starting with the first question out there, dioecious plants are, what do you mean by dioecious plants? Dioecious plants means they have separate sexes. For example, there will be a male plant and there will be a female plant. Female plant, male plant means man. Over here the flowers will be there which will be just having the carpel. Over here you will have the flowers which does have the, what do you call, the stamen. Apart from that, you do not have anything. Okay, that is what you mean by male and female plant. So what are the examples of dioecious plant? The example is papaya. Papaya. Okay, and now tell me the answer for this question. Autogamous only, both autogamous and gytenogamous, both gytenogamous and xenogamous, xenogamous only. What would be your answer for this? So Asya says it's D, Nagaraj also say it's D. So in dioecious plants, my dear children, you are absolutely correct. Okay, Hanumantreya also. That is, from here the pollen can move only to another plant. It cannot land on the same plant because there is no carpel to land on, right? There are only stamens out there. So it will definitely show only one thing, that is known as xenogamy, pure cross pollination, nothing else, pure cross pollination. Yes, Muskan also, you're absolutely correct. It won't be autogamous. Autogamy means from here to here. Suppose there is a flower like this. From here to here, if it was going on the same flower, you call this condition as autogamous condition. Now on the same plant, 2 different flowers, but on the same plant, if this is happening, that is known as gytenogamy. Anyway, both of them are not possible over here. Okay, so please be clear with this example. And there is also one more example of monoecious plants. That is castor, my dear children. What type of pollination do they show? Monoecious plants such as castor, what type of pollination do they show? So they do not show autogamy, but they show gytenogamy. Is it? Do they show gytenogamy? Read this example and let me know in the comment section. Okay? Castor. It's castor. So in them, what, my dear children, happens is that monoecious plants are the ones which will be having both the sexes in a plant. Now it can be like this. Suppose I have a female flower and then there is a male flower. They are unisexual flowers only one sex is there. Only one sex is there, but in a whole plant, if you see, there is male and the female. This condition is known as monoecious plant with unisexual flowers. Now there is one more condition where monoecious plants are there, but with bisexual flowers. The flowers has both the carpel as well as the stamen. It's same. Okay. So over here bisexual plants, but they are monoecious. Over here unisexual plants, but they are monoecious. So in this condition, one very important thing is there, that autogamy is prevented. Can autogamy happen over here? No. But gytenogamy will still be happening. Gytenogamy will be still happening. This is an example of outbreeding devices. In outbreeding devices, you studied about dioecious plants and also you studied about unisexual monoecious plant. Example was castor. Okay? Please remember this example. Hello Ravichandra. Hello Mohammed. Okay. Now going ahead with the next plant. Question number 2. Endosperm persists in mature seed in what? Endosperm will be still persisting in the mature seed in what? Groundnut and beans, castor and coconut, pea and castor, beans and pea. Yes, Nagaraj, in the previous question for monoecious plants like this, gytonogamy is possible, xenogamy is also possible, autogamy is not there. Okay, autogamy will not be there. You would be asked the definition, difference between gytogamy and xenogamy. Please prepare for that. I have explained in the complete revision video, the other one. Okay, the endosperm persists in, let me see, what are you telling me? So Hanumantraya says B, Muhammad Nihan says A, Nagaraj says is B. Okay, I have mixed answer for this question. Endosperm will still persist in only one condition, where it is monocot plant. And I'll tell you an easy way to understand this. Monocots are the one that cannot be broken down, the seed of which cannot be broken down to two parts. For example, wheat. Okay, for example, maize, for example, barley, for example, castor, for example, coconut. All of them, my dear children, they cannot be broken into two. Don't think that coconut, if I put like this, I can break into two. No, I'm talking about the seed out there. It cannot be broken down to two. So, it would be castor and coconut. Groundnut, you can break it down to two, right? So, because they are dicots. They are what? Dicot. Similarly, beans also dicot. Pea, you can break it down into two. That is also dicot. Castor is a monocot. Beans and pea again. So, your only answer that is correct is what? Castor and coconut. Always remember, in monocots, there is only one cotyledon. Okay, there is only one cotyledon. And there will be a little endosperm still left back. This is one very important thing that you need to remember. You know there is embryo. And embryo is made from the zygote with the help of nourishment given by the endosperm. So when it forms a dicot seed, it says that, embryo says that I do not need endosperm anymore. I am going to have my own what you call nutritive material, that is cotyledon. So this particular dicot would be having embryonal axis and two cotyledons. That is why it is dicot, two cotyledons. It would not have any endosperm left back because it makes its own reserve material. But in the case of monocots, they will have the embryonal axis and they will have cotyledon, only one cotyledon and little amount of endosperm. They will have only one cotyledon and also little amount of endosperm. So I hope this is clear. Okay. So your answer would be B. B. Fine. Chalo. Statement 1. In Viola, both autogamy and gytonogamy is prevented. Is it true? In Viola, both autogamy and gytonogamy is prevented. I'll make the Viola flowers. Now you tell me. Is autogamy and gytonogamy prevented in Viola plants? My dear child, Sonu, I think I have included at least 15 questions from each chapter. So we are on the third question from the second chapter and then we will go ahead with the next. Okay, before 7 we will finish it up because your chemistry ma'am will be there. Okay, chalo. Yes dear, it will be definitely helpful for you for tomorrow's examination. Okay, and I'll recommend you please watch the other video as well, that is the complete revision video if at all possible. Okay, the morning time you can just give a quick watch, but please read your NCERT. Okay, so over here, my dear children, autogamy will be there, pollen to the stigma of the same flower, autogamy is there. What about gytonogamy? Gytonogamy means between two flowers of the same plant. That will also be there. So here it is said that in Viola, both autogamy and gytonogamy is prevented. The answer is wrong. This particular thing is wrong. It is not prevented. It is still happening. Now Viola has two types of flowers. Always remember, one is the closed flower. You call them clistogamous flowers. And the other one are the open flowers, which are known as chasmogamous flowers. Chasmo means open, clisto, CL for closed. You can remember like that. In this condition, only autogamy will be happening. Gytonogamy will not be happening, but at least autogamy will be happening. Over here it is written both autogamy and gytonogamy is prevented, which is why this particular statement will be wrong. Now second statement, gytonogamy is similar to autogamy since pollen grains comes from the same plant. Gytonogamy is similar to autogamy because they have mentioned that since the pollen grains are from the same plant, that is why. So can I say it is genetically similar? The answer is yes, it is genetically similar. Geitonogamy is a type of cross-pollination, but it is not true cross-pollination. Only true cross-pollination is xenogamy. Here it's from the same plant, which is why there will be less amount of genetic differences also. Okay, please remember this is a very important question. So I'll say this statement is correct, this is false. So statement 1 is incorrect, but statement 2 is correct. Your answer would be D. Let me see who all got it correct. Hanumantraya and the Nagaraj got it correct. Asya got it correct. Let's see what about the other guys out there. Children. Very, very important thing, okay? Muskan also got it correct. Now, question number 4. Select the incorrect statement with respect to vegetative cell of the pollen grain. Select the, okay, Ravichandra also got it correct. Select the incorrect statement with respect to vegetative cell of the pollen grain, my dear children. What is vegetative cell? So if you talk about the pollen grain right over here, you have two cells. One is the vegetative cell, which is the bigger cell, and the smaller cell which is out there, it is known as generative cell. This generative cell floats in the cytoplasm of the vegetative cell. Now, it is bigger than the generative cell. Do you think the vegetative cell is bigger than the generative cell? The answer is, this is correct. You have to find what is incorrect, no? So, has abundant food reserve? Do you think it has abundant food reserve? Yes. So, someone has given me already the answer. Okay, Hanumantriya Nagaraj also says it's D. Asya says D. Okay. Has a large irregularly shaped nucleus? Yes, it has large irregularly shaped nucleus. That is also correct. Floats in the cytoplasm of the generative cell? The answer is wrong. Generative cell is a one that floats in the cytoplasm of the vegetative cell. And if you ask the shape of the nucleus, it is spindle shaped. In generative cell, it is spindle shaped. My dear children, how many layers does this pollen grain have? This is plasma membrane. Outside the plasma membrane, there is one very important continuous layer. It is known as intine. Intine is made up of two things. It is cellulose and pectin. Please remember, cellulose and pectin. Okay, then there is outer layer also, which is not continuous, it is discontinuous, because you see small pores out there. What do you call these pores as? You call these pores as germ pores. Okay, and this layer that you see over here, it is known as exine. It is known as exine. And this exine is made up of what? Very important thing. It is made up of the most resistant material out there, that is sporopollenin. Who's it? It is sporopollenin. It's the most resistant material. Now let's see. Okay, Hanumantriya, Nagaraj, Asya, Ravichandra, Saraswati Yadav, everyone says it's D. And children, I am just giving you extra knowledge. Suppose if at all you get questions around it, you can answer it. Now one homework question to you is that please go and see for the diameter of a pollen grain. Okay, diameter of a pollen grain. The second layer is exine and it is made up of sporopollenin. Okay, did I say something else? Okay, so children, I am just repeating. So over here, okay, that's all. You have to tell me the diameter of a pollen grain. It's what? 22250. Please go search for it. It's very important. Okay, now going ahead with the next thing, that is fifth question. Which one of the following statement is not true? Pollen grains of many species cause severe allergies. Is it true? This is true. Stored pollen and liquid nitrogen can be used in crop breeding program. Do you think? 25 to 50 diameter. Yes, that is correct. Okay. Micrometer in diameter, that is more correct. Okay, anyways, so coming ahead, my dear children, that's your homework. You can comment it down in the comment section. I'll be replying it to you there. Okay, now over here, stored pollen and liquid nitrogen, do you think it can be used for crop breeding program? The answer is yes. Okay, you can use it for crop breeding. Crop breeding as in you want to make superior varieties of plants, you can do that. You can take the pollen grains and then do pollination. Now, tape term helps in dehiscence of anther. Do you think tape term helps in dehiscence of anther? This particular statement is wrong. Why? Because you have certain layers out there. The outermost layer is epidermis, then comes endothecium, then comes middle layer. Okay, these three outer layers are the one which is responsible for dehiscence of anther. That means anther, for it to open and release all the pollen grains, that process is known as dehiscence. So for dehiscence, you need these three layers. The innermost layer, which is tape term, my dear children, always remember it is mostly binucleated in condition. That means it has two nucleuses in the cell. Okay, so over here this tape term is responsible for nourishment. Nourishment of what? Nourishment of the, what do you call, cells of the sporogenous tissue. That means the future pollen grains. So tape term is there for nourishment. Please remember it is in binucleate condition. So exine of pollen grain is made up of sporopollen, that is also correct. Intine is made up of pectin. Okay, someone said pectin, so it's wrong. Intine is made up of pectin and cellulose. Exine is made up of sporopollen. This is absolutely correct. And over here C is your answer. Muskan, Ravichandra, Asya, everyone was there. Okay, Akshita, great. Okay, Saraswati, everyone, kudos to you, my dear children. Now going ahead with the next question. From the statements given below, choose the option that is true for a typical female gametophyte of a flowering plant. What would be the answer for this particular question? Akshita, 5 marker, my dear child, I have made a separate video on 2, 3 marker, 2, 3 and 5 marker. Okay, can you please watch that? It's there in the YouTube channel itself. Okay, I made a few days back. That has all the 2, 3, 4, 5 marker questions out there. You can watch it, not 4 marker, 5 marker, okay? Now tell me the answer for this question. From the statements given below, choose the option that is true for a typical female gametophyte of a flowering plant. It is 8 nucleate and 7 celled at maturity. Is it talking about the female gametophyte? That means you're talking about the embryo sac. Do you think it has 8 nucleus but 7 celled? Yes. So over here, 1 cell, the second cell. These cells are known as synergids. Okay, and over here one more cell, that is the egg cell. Over here 3 cells, 1, 2, 3. These are known as the antipodal cells. Now this big thing that you see over here, this is the central cell with 2 nucleus. And these nucleus are known as 2 polar nuclei. So how many cells? 1, 2, 3, 4, 5, 6, and this one is the 7th cell. But how many nucleus? 1, 2, 3, 4, 5, 6 and 7, 8. 8 nucleus is there. So this is correct. Okay. Now it is free nuclear during the development. That means what? Basically, when the embryo sac is formed, no, it is not formed as such. There is one nucleus which undergoes division, divides into 2 nucleus. Then again this nucleus divides into 2 more nucleuses. Again it goes like that. 3 divisions will be there. 3 mitotic divisions will be there. Okay, and in this condition it's free nuclear. What do you mean by that? Immediately it is not surrounded by any membrane out there. It is not surrounded by any cell wall. It stays in that condition, but later it's slowly surrounded. It is getting slowly surrounded by membrane. So it is known as free nuclear condition. So this is also true. It is free nuclear during the development. Yes, Ranjit, tell me. Oh, you are so cute. Oh, thanks. Okay, now it is situated inside the integument but outside the nucellus. Is it true? Is it situated inside the integument but outside the nucellus? Ma'am, I do not know where is the nucellus. So over here, listen. So this is the ovary, my dear children. And ovary, over here you have the placenta, which is responsible for nourishment of the ovule. Okay. And inside the ovule, my dear children, you have certain cells like this. These cells form the cells of the nucellus. Cool. Now over here there is placenta to nourish this guy. What is this guy? This is the ovule. And how is it joined? It is joined with the help of something that is known as funicle. And what is the attachment spot? This is known as hilum. And the ovule feels very cold inside this. So you have 2 integuments. One integument and the second integument. You see, I have kept an opening. What do you call this opening as? You call this opening as micropyle. Okay, now always remember the opposite end of micropyle will be known as chalazal end. Will be known as the chalazal end. Suppose the placenta is here and the funicle is here. Where is the chalazal end? Chalazal end will be still here if the micropyle is on this side. Please understand that. Now here I said there is cells of nucellus. Apart from that, there is one very important cell, that is megaspore mother cell. What is it? Megaspore mother cell. This megaspore mother cell will undergo division, form one functional megaspore. Okay, and this one functional megaspore will form the embryo sac. So now you tell me, now the embryo sac is over here. It is situated inside the integument, is it? Yes. But outside the nucellus? No, it is inside the nucellus cell also. This thing that you see is your embryo sac. So this statement is wrong. Now it has an egg apparatus situated at the chalazal end. So my dear children, over here, wherever you have the egg, this end is known as micropylar end. And the opposite end will be chalazal end. So here they are saying the egg apparatus is situated at the chalazal end. The answer is wrong. Egg apparatus means synergids and the egg. All of them are towards the micropylar end, and this should be a wrong statement. And who is the one that will be helping the pollen tube to come inside this particular thing? It is the filiform apparatus which is present on the synergids. Always remember, filiform apparatus present on the synergids, which with the help of which it will guide into this particular egg cell that you see. Okay, and while doing it, one of the synergid, they get degenerated also. Remember that also. Okay, will you forget it? I hope no. Okay, please don't forget it. So, what would be your answer? Choose the option that is true. Your answer would be A and B. So, answer is C. Sagar, hello. Akshita was answering Hylam. Okay, I saw it now, but yes, let me see who all got it correct. Asya got it correct. And then Hanumantraya got it correct, Asya, Sagar got it correct, Nagaraj got it correct, Saraswati also got it correct. Kudos to you. Now going ahead with the 7th question out there. Tell me this. Identify the correct sequence of arrangement of cells in the mature embryo sac from the micropylar end. From the micropylar end, you have to tell the arrangement. I just told you right now. Okay, how is it arranged? Tell me. So this one is a synergid, then you have egg, then you have central cell, then you have antipodal. Okay, so what will be the answer for this particular question? Come on children, answer for 7th question. Put it like this, 7 A, B, C, D. What is your answer? 7 A, B, C, D. What would be your answer? A. Mansa says it's A. Asya says it's A. Okay, Saraswati also A, Nagaraj also A, oh my god, Sagar also A. So yes, the answer is A. First synergid, then egg cell, then central cell, then antipodal from the micropylar end to the chalazal end. Please remember there are certain examples given in your NCERT. Okay, in the box of before the megasporogenesis there is a box and stuff, sorry, before the structure of megasporangium there is a box where they have mentioned about syncarpus, apocarpus condition. Okay, please go through that, it's very important. Syncarpus means all the carpels are together. Where do you see it? You see it papa where? Okay, apocarpus means all of them are different arranged. And the example is Michelia. And there is a ovule which is ulta, anatropous ovule, where what is happening? The chalaza, sorry, the what do you call, what is this? Placenta as well as the funicle which is right over here, it will be over here. Ulta condition will be there, that is anatropous ovule. So please go through all the pictures and the examples, it is going to help you really well. Okay, now even in the absence of pollinating agents, seed setting is assured in. That means even if pollinating agents are not there, still you will be able to find seed. Where? Question number 8. Even if pollinators are not there, still you will be able to find seed. Hanumantraya says it's C. Is it C? Okay, that's cool. Cucurbita, papaya, commelina, maize. Now only in one condition you would not need pollinating agents. Only when the flower is closed and when you have both the stigma as well as, what do you call it? You have both the carpel as well as stamen inside, right? What is this condition of flower? This is known as clistogamous flowers. And where do you find it? In 3 condition. In 3 condition, my dear children. Okay, sorry, in 3 flowers, sorry. One is viola, one is oxalis, another one is commelina. So your answer would be C, commelina. Okay. So Asya, A is wrong, Manasa, A is wrong, my dear. So, okay, you're telling about 7. Sorry. 8th question please. It would be C, that is commelina. Okay. Spurti is back again. I hope that's clear. Commelina, oxalis or viola, either of the options if it comes, you can go for it because they have clistogamous flowers. Where do you find the closed flowers? Towards the root region, very important question, or the rhizome region. Okay, root or rhizome region. And the open flowers would be in the egg cell position out there. Now, yes, Manasa, you're also correct. Now, 9th question. Select the correct sequence of anther wall layers. Anther wall layers, I told you 2E comes first. E, then MT. You know EMT, ear, mouth, throat, doctor, EMT doctor. right? Seamless, that is ENT actually, but anyways, you can remember like that as of now. So E stands for epidermis. Epidermis is always outside. Inside that, endo, and then middle, and then the tapetum. Okay, so your answer for this question would be epidermis endothecium. B. Okay, so let me see who all got it correct. Children, if you're not giving me the question number, I won't be able to understand what question are you answering. Nagaraj says 9, option B. Okay, Asha says it's C. Manasa says it's B. Yes. Children who got it wrong, can you please check it once more? Okay, check it once more, okay? Tapetum, always remember it has multiple nucleus. Because karyokinesis has happened, the nuclear division has happened, but cell division didn't take place. Cytokinesis didn't take place, karyokinesis took place. Okay? Now, which of the following is not a correct statement? Tell me first. Placenta is located outside the ovarian cavity. Is the placenta located outside the ovarian cavity, my dear children? So, this is ovary, this is your ovule, this is the ovarian cavity. Placenta is over here itself. So, this is wrong statement. Okay, central cell develops into PEC, primary endosperm cell, after fertilization. Is it true? Yes. So, 2 fertilization take place, double fertilization. 1 fertilization with the male gamete and the female gamete, that forms the zygote. The second fertilization with the male gamete and the central cells, the 2 polar nuclei of the central cell. That is known as triple fusion. And what does it form? It forms primary endosperm cell. What nucleus does it form? Primary endosperm nucleus. What is the ploidy of it? You should know that. What is the ploidy of primary endosperm cell? The primary endosperm's ploidy is 3N. Triploid. Why? Because you have fused 1 male gamete and 2 polar nuclei. So here it is N, here also it will be N and N. 2 nuclei are there, no? So N plus N plus N is 3N, triploid. The only thing that is triploid in nature there. Now what is the ploidy of a zygote out there? It will be diploid. N from the male gamete, N from the female gamete, 2N. Okay, that is diploid. So over here, central cell develops into PEC, that is correct. Orchid contains more than 1 ovule in an ovary, correct. Egg apparatus consists of 1 egg cell and 2 synergid cell, that is also correct. Okay, egg apparatus consists of 1 egg cell and 2 synergy cell, that is also correct. So only wrong answer is A. Okay, children, know about triple fusion, syngamy and all of it. Okay, very important. Give and remember the examples, they can give you example related questions. Now question number 11th out there. Select the correct sequence of events in microsporogenesis. What happens? You have sporogenous tissue. Inside the sporogenous tissue, what is there? So I got some answers. Manasa said option A, Nagaraj said A, Asya said A. Kudos to you, and Nagaraj also said diploid, triploid, everything. Kudos to you. Okay. Now over here, you have to tell the correct sequence of microsporogenesis. 11th A. Hanumantraya says it's A. So my dear children, you have sporogenous tissue. Inside the sporogenous tissue, it is made up of so many. Unlike the females, in the males you have so many microspore mother cell. There it is megaspore mother cell, MMC, but here it is microspore mother cell. This microspore mother cell, which is having 2n number of chromosome, undergoes division. What division? Meiosis, because you have to form the gametes, right? So over here, meiosis 1, you get 2 cells. Again this divides, you get 4 cells at the end. What do you call them? Microspores. But you know what? They are all together. So you call it microspore tetrad. 1, 2, 3, 4. 4 of them together. So microspore tetrad. Later, when the anther gets dehydrated, they will all separate. And these microspores are the one that will form the pollen grain. Pollen grain is a gamete. Is it a gamete or not? Tell me the answer in the comment section. Pollen grain, is it a gamete or not? Or is it a gamete of fight? What is it? What is the difference between that? My dear children, pollen grain, the question is it a gamete? The answer to the question is no, ma'am. It's not the gamete. It is the one that will be helping in the formation of the gamete. Pollen grain has 2 cells, vegetative and generative cell. Remember that. So it is the male gametophyte. Okay. So over here 4 of them are involved in pollen grains. Sporogenous tissue, microspore mother cell, then microspore tetrad, then microspores. Important thing that you need to revise from your end is what? It is the one that will be forming the gametes later on. And always remember, as I told you, there is one vegetative cell and one generative cell. Usually in all the flowering plants, this goes like this only, in two-celled condition. The pollen grains are released in two-celled condition. How much percentage? 60% of angiosperms, most of the angiosperms. But there are certain flowering plants where this generative cell will divide into two. That means 2 male gametes. Then in total how many cells are there? One cell is vegetative cell, the other cell is this cell, the other cell is this cell. That is 3-celled stage. How many of them? 40% of them. 40% are released in 3-cell stage. 60%, more of it are released in 2-cell stage. After going there, it will divide later on. Okay. Got it? Chalo. Going ahead. Question number 12th. Children, I hope you got it. I hope this is clear to you. Yes. Now over here, question number 12. Statement 1 says apomixis is the production of seeds from unfertilized ovules. Is it correct? This is a very important topic. Apomixis is the production of seeds from the unfertilized ovules. The answer is correct. Okay. Why the answer is correct, my dear children? Whenever seeds are produced without fertilization, you say that this process is happening, which is known as apomixis. Whenever fruit is produced without fertilization, you say what is happening? Without fertilization, a fruit is producing. What do you call this? This is known as parthenocarpy. Okay, so over here, this statement is absolutely true. You have to tell about the next statement. Embryos produced from apomictic seeds are not identical to the parent plant. Do you think when seeds are produced without fertilization, my dear, in fertilization both the male and the female gametes are coming together. In that case, you see that the new plant that will be coming out, it will have variation. But in this condition where there is no fertilization, my dear children, do you think there will be variation? No. And if there is no variation, don't you think the baby plant that is going to grow, my dear children, it will be just like the parent plant? Yes. So over here, ulta statement is given. Embryos produced from the apomictic seeds are not generally identical to the parent plant. That is again wrong statement. This is wrong. So first statement is correct. Second statement is wrong. Statement 1 is true. Statement 2 is false. Okay. So your answer would be this, A. Your answer is 12A. Sagar, Nagaraj. Okay, Manasa, you got it correct. Hanumantraya, just check it once, okay? Now, the ploidy of perisperm present in beet seed is. The ploidy of perisperm present in the beet seed is what? Haploid, diploid, tetraploid or triploid? The ploidy of perisperm. Any guesses? Children. Come on. It's soon gonna finish. What is perisperm? Perisperm is basically, suppose you have the ovule and over here you have the cells of the nucellus. So usually when embryo sac is formed from that one megaspore mother cell, it forms the embryo sac. My dear children, what happens? This cell usually degenerates away. But somewhere it can still be there and it will be helping in growth of the future embryo out there. So this persistent nucleus cell which is there, it is known as perisperm. It is known as perisperm and it's just like a normal cell. It's normal cells which come together to form the perisperm, right? And normal cells, what is their ploidy? It is 2N, diploid in nature. Only gametes have half of it. Only PEC has been triploid. So rest of the cells are 2N in nature. So my dear children, over here, your answer would be diploid, B. Okay? Cool? 13B, Asya said B, Nagaraj said B, Sagar said B. Okay. Sagar is asking, are you ready for chemistry test? I hope children you are ready. Well, you're getting a live really soon, but before that be ready for your biology test also. Okay. Now the correct sequence of embryonic development in angiosperm is what? Chalo, fast. The correct sequence of embryonic development in angiosperm. Tell me the sequence. Zygote, then what? The zygote is formed, the zygote has to form an embryo. Embryo has different stages. First one that is formed is pro-embryo. It does not know what to do. Okay, it is pro-embryo. And then later on, it forms globular embryo, round-shaped. And then heart-shaped embryo. Then what happened, my dear children? It forms mature embryo. Okay, it forms mature embryo. So first is, always remember you have zygote. After zygote, you have pro-embryo. Okay, pro-embryo becomes globular embryo. Okay, it's like we are growing. When we grow, we gain a little bit of weight, no? Then we have some crushes out there, the heart-shaped embryo, and then it becomes mature, the heart is broken, right? Similarly over here, mature embryo is there. So your answer would be C. Please remember that. You have to remember this particular thing importantly. You might forget, but I don't want you to forget it. I don't want you to forget it, that's all. So your answer is C. Let me see who all got it correct. 14 C, Nagaraj said it's C, Skill said it's C. Skill initially said it's D also. Abba. Okay, chalo. My dear children, don't get it wrong, okay? Fine. Next, okay. The next chapter is human reproduction, but before that, one very important thing about this chapter, that is the megasporogenesis. No, the megasporogenesis, always remember there is one megaspore mother cell, divides into two in meiosis 1, again divides into 2 in meiosis 2. Now, only one will be surviving up there to form the, what you call the functional megaspore. The functional megaspore. Rest all of them will get degenerated. Always remember that. Only one stays there. And this is the one that forms the embryo sac, which is why you call this as monosporic development. Monosporic development. Okay, only one spore is developing to an embryo sac. And now, suppose this functional, functional megaspore is there. It will have to undergo division. How many division I told you? 3 mitotic division. Okay, this nucleus divides into 2, again undergoes division, and again will be undergo division. Thrice it undergoes division. What division? Mitotic division. All these things are really important. Okay, and also one very important thing. What is the, example of perispermic seeds? Beet and black pepper. Okay, B square. Remember all these things. Now, going ahead with the next thing out there, you have the next question from human reproduction. The first question is, scrotum helps in maintaining the temperature of testis, which is A, lower than the normal body temperature. Each of the testes has about B compartments and measures C centimeter in length. Select the option that fills the blank correctly. Come on, children. What is the answer for this question? Scrotum maintains how much temperature lower than the normal body temperature, my dear children? It will be 2 to 2.5 degrees Celsius. How many compartments are there in the scrotum? 250 compartments are there. 250 testicular lobules are there. Now, what is the length? It is 4 to 5 centimeter in length. Okay, please remember it. So, your answer would be D. What is the width? It is 2 to 3 centimeter. 2, 3, 4, 5. Remember like that. So, width is 2 to 3 and the length is 4 to 5. Okay, and 2 to 2.5 degrees Celsius lower than the normal body temperature. Otherwise, spermatogenesis will not be happening. So, Nagraj says is B, Sagar says is B, Manasa says it B. Okay, cool. Now, going ahead to the next question out there. The function of myometrium layer present in the uterine wall is 2. What is the function of myometrium? Myometrium is the muscular layer. Formed of what? Formed of the smooth muscles. What is its role? Is its role to undergo cyclic changes during menstrual cycle? No. That is the role of endometrium. That is the role of endometrium. Is its role to exhibit strong uterine contraction during parturition? Parturition means expelling of the baby. So, is it that? Yes. Myometrium is for contraction during parturition. Give protection to the uterus. Is the function of myometrium that? No, that's the role of perimetrium, the outermost layer. Now, help in implantation process. Implantation means embryo comes and gets implanted into the wall. Wall is what? Endometrium, not myometrium. So, myometrium's only role is to what? Undergo strong contractions. Okay. Just a second, children. Yeah. Live. Okay. So your answer would be B. Okay. Now going ahead with the third question. Does this live chat include 2 and 3 marker question practice 2? Dear Ankita, so I had made a separate video on complete revision where I have spoke about the 2 and 3 markers separately. If you want to watch that, you can watch that. There are 2 marker, 3 marker, 5 marker, everything is there. But I'll recommend you to be live over here as well for asking doubts. And if at all you see some new questions, you can solve them as well. Okay? So seminal plasma in human males, that you can watch in 2x speed. It will be really good after a quick NCERT read. If you watch that video, it will be helping you well. Now seminal plasma in human males is rich in what? Fructose and calcium, glucose and calcium, DNA and testosterone, ribose and potassium. Seminal plasma, my dear children, is made up of 3 secretions. Secretions from the prostate, okay, secretion from the seminal vesicle, and secretion from the bulbourethral gland, that is also known as the Cooper's gland. Okay, seminal vesicle will be giving something that is known as fructose so that the sperms get energy. Prostate will be giving 2 things, one is calcium and the other thing is clotting enzymes. Okay, and bulbourethral is responsible for lubrication. So all of them together gives what? Seminal plasma. Seminal plasma plus sperm will be forming the semen. So over here your answer is fructose, A and calcium. Yes, you are absolutely correct, Nagaraj, Sagar, Manasa. Okay, the answer is 3A, fructose and calcium. Now next question, statement 1. The process of release of sperms from the seminiferous tubules is called spermatogenesis. The process of release of sperm from the seminiferous tubules is not known as spermatogenesis. It is known as aviation, spermiation. Is it? Is it spermiation? Is it spermiation? Yes. This one would be spermiation. The process of release of sperms from the seminiferous tubule. You can remember like that. Okay, it's easy way to remember. Now, the spermatids are transformed into spermatozoa by the process called spermiogenesis. Is it true? Is it true? So, you get, for example, you have the spermatogonia, the sperm mother cell out there, yeah. Spermatogonia, some of them divides and again forms spermatogonia out there. Some of them they differentiate, they undergo mitosis and they differentiate into the primary spermatocyte. Primary spermatocyte. Now this primary spermatocyte undergoes meiosis 1. You get 2 cells. Here it was 2n, here it becomes nn. This is secondary spermatocyte, my dear children. Now this secondary spermatocyte again undergoes division into 4 cells out there, all of them having half number of chromosome. These are known as spermatids. They are like kids, they are immature, they are globular in shape. And now my dear children, they undergo a process which is known as spermiogenesis. Spermiogenesis, when they gain their shape like this and they form a structure which is known as spermatozoa. From spermatids, that is the kids, you get the spermatozoa. So your answer for this question would be statement 1 is incorrect. Why? Because it is spermiation, but statement 2 is correct. So who all got it correct? D option. Let's see. Sagar got it correct. And then Nagraj got it correct, Manasa got it correct, Hanumantraya also got it correct. Kudos to all of you. Children, are you understanding? I suppose you are understanding. If at all you are not, please ask me the questions. Okay? Ma'am, what is spermatogenesis? Spermatogenesis is the whole process that we just discussed. From here to here, the whole process of formation of sperm is known as spermatogenesis. From here to here, it is spermiogenesis. Now, when the sperms are moved out of the seminiferous tubules, you call it spermiation. Okay? Now, to produce 2000 sperm and 400 ova, how many spermatogonia and oogonia are required? To produce 2000 sperm and 400 ova, how many spermatogonia and oogonia required, my dear children? So from each spermatogonia that you saw, my dear children, one primary spermatocyte was being formed. So we can say that from one primary spermatocyte, if you roughly calculate, you get 4 sperms. So this spermatogonia which was there, it was differentiated into the primary spermatocyte. So let's count it as just one. Okay, you don't need to worry. So if suppose it was 2000 spermatogonia, then 2000 into 4, which is what, 8000 sperms will be formed. But as of now the condition is ulta. Over here they are saying there are 2000 sperms. There are 2000 sperms. How many spermatogonia are there? So instead of doing into, you will do divided by 4. So what is it? The answer would be 500. 500 spermatogonia is there. What about ovum? So over here, from one oogonia, my dear children, oogonia also same undergoes differentiation, forms one very important thing, that is known as primary oocyte. Primary oocyte. This primary oocyte will undergo division. Okay, one cell will be small and it will get degenerated. This is known as the first polar body, and this one is known as the secondary oocyte. So it does not undergo division, it stops there. This one will undergo division, that is meiosis 2, and over here again this one gets degenerated. This is known as the second polar body. Over here you just get one thing at the end, and that is the ovum. So you started with 2n, this is the primary oocyte, 2n. You got n and n, here also n and n. Haploid ovum is produced, but only one. So from one ova you get only, from one oogonia you get only one ovum. So if 400 ova is there, so how much oogonia will be there? It will be 400 oogonia only. So your answer would be 500 spermatogonia and 400 oogonia. So always when it is sperm, you have to divide by 4. Okay, if your sperm's number is given. So I hope this is clear. So yes, Hanumantraya, Sagar, Manasa, all of you got it correct. Kudos to you. Now question number 6. The release of sperm from the seminiferous tubules is called. What do you call this process? We just discussed this. The release of sperm from the seminiferous tubules. What do you call it? Release of sperms from the seminiferous tubule. Sagar says it's A, Hanumantraya says it's A, Nagaraj says it's A. Okay, Nagaraj is back again. 6A. Anyone else out there? The answer is A, spermiation, aviation. Okay, remember like that. It will be easy for you. Now question number 7. What is the correct sequence of sperm formation? Again we discussed this thing. What is the correct sequence of sperm formation? First thing that you have is what? Okay, Manasa also said it's A. What is option C meaning, ma'am? Spermiogenesis, baba. Spermiogenesis we studied, no, spermatids to what? Spermatozoa is spermiogenesis. Okay. Now here correct sequence, please of sperm formation. Spermatogonia, spermatozoa, spermatocytes, then spermatids. What will be the answer? First thing that you have is spermatogonia, so this option you can cancel it off. Then from spermatogonia you get the primary spermatocytes. Then from spermatocytes you get spermatids. The kids get mature, you get spermatozoa. Okay, so your answer would be B. Children get it confused from this and this. I already told you. TIDS matches with kids also. So kids come first and then it becomes mature. Spermatozoa is a mature one. So keep it at the end. So your answer would be B. Your answer would be B. Okay, Hanumantraya, Sagar, Manasa. I hope that's clear. And you have got it correct. Kudos to you, children. Now question number 8. Which among the following has 23 chromosomes? That means you are asked about haploid number of chromosome, half the number of chromosome. Usually we have 46 number of chromosomes, right? But which or one among them has 23, half the number of chromosome? What would be the answer for this? Spermatogonia, it will be a normal cell. Okay, it is 2n in nature, it is not half. Zygote is formed when male and female gamete comes together, n and n, 2n, it is also diploid. Secondary oocyte, my dear children, it is formed after meiosis 1. From primary oocyte, when it undergoes division, you get secondary oocyte. So it is half, it is containing half the number of chromosome. What about oogonia? Again, oogonia is something that will be having 2n number of chromosome. So over here your only answer is C option, secondary oocyte. It's C. So who all? Hanumantraya, Nagaraj, Manasa, Sagar, all of you got it correct. Kudos to you. Now question number 9. Match the items given in column 1 with those in column 2 and select the correct option given below. Column 1 and this is column 2. Okay. So proliferative phase. What is this phase also known as? Proliferative phase means something is getting proliferated, that is your endometrium. Slowly starting to getting reformed. It's not fully formed, it's just starting to getting reformed. In this phase, one more thing happens. Follicles are developing, follicular phase. When the follicles are developing, they release something that is known as estrogen. Please remember that diagram of estrogen, progesterone as well as FSH and LH. If at all you do not know, please watch the complete video. It is going to help you really well. Okay, so the follicles, when they generate under the influence of FSH and LH, one very important thing is being released, that is estrogen, which makes the endometrium also reform a bit. Okay, so A goes with 2. A goes with 2. I just have one option. With that I got the answer. Secretory phase, it is also known as the luteal phase. Now once follicular phase, the follicles is generating, my dear children, one point comes when the amount of LH is way too much, and what will happen? The follicles will, the graphene follicle which is there, it is rupture and it will release what? These oocyte, the secondary oocyte, and whatever is left over in the graphene follicle, it will form the corpus luteum. This phase is known as luteal phase, and during this phase the corpus luteum releases one very important hormone, progesterone, along with estrogen. So progesterone again maintains the endometrium, makes it thicker, and also make it release secrete some very important nutrients if at all the embryo comes and lands. That's why. So it is also known as secretory phase. Menstruation is basically when the breakdown of endometrial lining occurs. Okay. Hi anime. So your answer would be 2, 3 and 1. Okay. C is the answer. Going ahead with the next thing. Question number 10. Which of the following hormones is produced only during pregnancy by the ovary? Which is the hormone that is produced only during pregnancy by the ovary, my dear children? What will you answer for this? Only during pregnancy, okay? And that is released by ovary, not by placenta. I'm not talking about placenta. Please remember the placental hormones also. You have 4 of them, HCG, human placental lactogen, and then what? You have estrogen and progesterone. Apart from that, you're talking about ovaries hormone. So there is one hormone which is known as relaxin. It is known as relaxin. D. Okay? Fine. Why do we need relaxin? For the cervix to relax so that the baby can easily move out. During later phase of pregnancy it is released. Okay, not during initial time, during later phase. So yeah, I hope you understood it now, Sagar. But why? Okay. Now choose the correct option from the table given below for the formation and dissolution of the labeled part in the given diagram. What? Ma'am, you know Goku? Noda, I do not know. Okay, so over here you have to tell me what helps in the formation of this particular labeled part, that is zona pellucida. What is that something that forms the zona pellucida? It is the secondary oocyte. So the one that has primary oocyte, we will delete it up, and the one that has secondary oocyte, we will keep it there. And dissolution of the labeled part, the one that is responsible for breaking down the labeled part, what is that? So secondary oocyte is the one that is forming the zona pellucida, but the sperm, when it comes to fertilize the egg, my dear children, so before fertilization, when the sperm tries to enter the zona pellucida, my dear children, that's when it gets dissoluted. So I can say before fertilization, but this is actually a kind of what do you call, a twisted question. Okay, why the question is twisted is that, my dear children, if you talk about complete dissolution of zona pellucida, it will be done after fertilization. When the zygote is formed, completely the zona pellucida will slowly go off later on. Okay, will go later on. But as of now, since you do not have the concept being taught, we can just say, and they have not mentioned complete dissolution, we can say before fertilization itself, when the zona pellucida, when the sperm comes in contact with zona pellucida, it will try to remove certain part of it and get inside the egg. Okay, so which is why your answer would be C. Okay, cool. Hanumantraya and Sagar, you got your answers correct. Now, question number 12. During pregnancy, the fetus develops limbs and digits. After how much time? During pregnancy, the fetus develops limbs and digits. After how much time? After 3 weeks, after 8 weeks, after 6 weeks, after 9 weeks. Always remember, you have to remember the timeline. After 1 month, you get the heart beating sound. After 2 months, you find the limbs, the digits being formed. After 3 months, all of this, okay, all of the above, plus organ system, plus organs are developed, plus external genitalia is formed. External genital organ is also being formed. Okay, and 4th month is skipped in your NCERT directly into the 5th month. Movement, the first movement of the baby is seen over here, plus some hair on the head, only on the head, okay, not on the body. On the body it will happen in the 6th month. Hair on the body, eyelids are separated, eyelids are separated and all of it. Okay, ma'am, I have chemistry exam tomorrow, is it? Okay, you will get a class at 7 o'clock, be there, okay? Now, so over here, during pregnancy, the fetus develops limbs and digits after how many months is being asked? No, they are asking about the weeks. How do you calculate the week? In 1 month, you have got 4 weeks. In 1 month, my dear children, you have got 4 weeks. So, in 2 months, how many weeks will be there? 2 into 4, it will be what? 8 weeks. 8 weeks. So, after 8 weeks, you will get your limbs and digits being formed. Okay, now 3 into 4, 12. After 12 weeks, you will be having your heart, limbs and digits, organ and external genitalia. This is the first trimester, the first 3 months. Okay, now after 6 months, that is the second trimester. First 3 months and the second 3 months, that is the second trimester. After second 3 months, you will have everything that you see over here. Okay, cool? Chalo. Now we are going ahead with the next chapter, that is reproductive health, and all, as I told you, all the other questions, the 2 marker, 3 marker, 5 marker that you're searching for might be there in the complete revision video. Please watch it if at all needed. Please read your NCERT first and then go ahead by solving questions. Don't be stuck at your NCERT. Go ahead, solve questions, more and more questions. Okay? Is it okay? Children, are you understanding what I am trying to teach? If you are understanding, give me a like button. Click on the like button right now. Subscribe. Also share it with your friends. And tomorrow also I think there's a live for children who are having biology exam day after tomorrow. So if you are someone like that, you can ask them to join. I'll include more different questions out there. Okay? And from this chapter, again, some important things. Please know about all of the months. Please know about the hormones secreted by the placenta, 4 of them. HCG, human chorionic gonadotrophin. HPL, human placental lactogen, estrogen and progesterone. There are certain hormones which get increased in many folds in the blood only during pregnancy. That means they're already there, but they get increased. What are those? Those are cortisol, thyroxine, prolactin, all of it. Okay, thyroxine, cortisol, prolactin, all of these things are increased in many folds. If the term comes many folds, just know this is the answer that you have to give. What is the importance of it? Maintenance, metabolism, all of it. Okay, why is parturition known as neuroendocrine mechanism? You have to tell that there are certain receptors that get active, stress receptors, and they give signal to maternal pituitary pituitary gland releasing oxytocin. Now, hormone comes into play. All of this thing is important. Again, I have explained in the complete revision video. Yes, da, I know Ronaldo. Okay, okay, chalo. Chalo. Reproductive health. So, apart from that, yeah, okay. Chalo. First question, decreased MMR and IMR and increased people in reproducible age in a population will result in. MMR means what? MMR means maternal mortality rate. Okay, anime style, I want to ask you a question. Do you know MMR and IMR? If you know, answer this question. Okay? Chalo. First, decreased MMR means maternal mortality rate. So, if mothers are not dying, okay, it is getting decreased, so it's a good thing. Infant mortality rate is also getting decreased, okay, and there is an increase in people in reproducible age. Then in this condition, do you think growth rate will increase or decrease? The answer is it is going to increase because everyone is alive and everyone is growing and there is more people in the reproducible age, my dear children. So your answer would be A. Okay, your answer would be A. Going ahead. Children, I need participation. Come on, this is the last chapter that we have. We're going to finish it really soon. So, maximum amount of participation, maximum output, maximum doubts. Cool? Chalo. Which of the following is an indicator of reproductively unhealthy society? Unhealthy society, okay? Increase in infant mortality rate. It's not good, right? Children are dying. So, this is what? An indicator of reproductively unhealthy society. Decline in maternal mortality rate. No, it's a good thing. It's a good thing. So, that is not an unhealthy society, right? Now, widespread awareness of contraceptive methods. That is also a good thing. detection and cure of sexually transmitted diseases, that is also a good thing. The only thing that is not good over here is increase in infant mortality rate, and that would be what do you call, indicator of reproductively unhealthy society. Okay, got it? Going ahead with the next question, question number 3. Children, if you do not answer, this particular session is going to be really boring. So I want maximum participation. Sagar, come on. Sagar said A is the answer. Okay, now I saw the answer. Now you have question number 3. Amniocentesis. Try this one, all of you. Amniocentesis is a medical procedure where some of the amniotic fluid of the developing fetus is drawn to analyze the fetal cells and dissolve substances. By this term, when you read this, you will be like, oh my god, what I am reading. So I'll explain you. So this is the amniotic sac where the baby will be there, my dear children. This is the baby out there, and over here there is a fluid which is known as amniotic fluid. Okay, now the cells of the baby are shut down over here in this particular fluid. Okay, so this cell will be having nucleus will be having genetic material, and by examining the genetic material we can find out what's wrong in the genetic. Do the child have some genetic disorders, stuff like that, right? So what do people do? They take in the fluid like this and they examine the cell. It is actually used for finding genetic disorders. You should be knowing the name of the genetic disorders, even though you might have not studied this until now. Okay, that chapter might have not been completed, like principles of inheritance and variation. So there are certain diseases such as, what do you call, what was it? Hemophilia, sickle cell anemia. Okay, hemophilia, sickle cell anemia, which you can find out with the help of amniocentesis. But people have started misusing it. Why? For determining the sex of the child, whether it's a female child and the male child, because anyway you get to see the chromosomes, right? So with the help of that, you are determining the sex of the child, and female feticide was increasing, which is why amniocentesis was banned. Now, it was banned in order to stop female feticide. Which of the following highlights the importance of amniocentesis despite the ban? Despite the ban, there is some importance out there. What is it? Herpes is not, herpes, my dear child, not categorize it as a genetic disorder. Okay, it's a sexually transmitted disorder. Now over here, identification of decline in sex ratio, no. You have to find what's the good thing about it. Detection of genetic disorders. This is the only good thing about amniocentesis. You can find it and it will be sorted out. Okay, not sorted out. Basically, if at all the child is not healthy, then there is no point of bringing it into the world. It probably, if it is very risky to the mother as well as the child, the parents can go ahead with MTP also. Okay, only in this condition. Okay, fine. Now, so the answer is D. I hope that's clear. Okay, and the examples given in your NCERT is hemophilia and sickle cell anemia. Fine. Now, statement 1 says lactational amenorrhea is the absence of menstruation during intense lactation. So during menstruation, when the mother, during lactation, when the mother is lactating, when the mother is breastfeeding, do you think there is absence of menstruation? The answer is yes. Why? Because in our body, there is a hormone that releases too much, that is known as prolactin. It says that FSH, LH, please stop your secretion. So they do not have FSH and LH. Without FSH and LH, there is no ovulation, there is no menstruation, anything happening out there. So lactational amenorrhea is the absence of menstruation during intense lactation. You're absolutely correct. This is highly reliable method of contraception. Do you think it's a highly reliable method of contraception? The answer is no. Why? Because there are cases where, my dear children, menstruation still can happen at times. So it is not a reliable method. And if menstruation is happening, that means ovulation might have also happened. So if some couple have sexual intercourse during this particular period thinking that they might not have a baby might end up getting a baby. Okay, which is why it is said that is highly unreliable. Okay, so over here it is written it is highly reliable. The answer is it's not. This is not highly reliable. So only one statement is correct. Statement 1 is correct, and the statement 2-1, which is C, is incorrect. C is the correct answer. Manasa and Sagar. Yes. Okay. Going ahead. Statement 1. IUDs are ideal contraceptives for females who want to delay pregnancy or space children. Yes. The answer is yes or no. IUDs means intrauterine devices, the ones that you put inside the uterus. Okay, so they are the ideal contraceptive. In your NCERT it is written they are, and it is also written they are one of the most widely accepted thing out there, method out there. That is also correct. And IUDs are specifically for spacing children. So for example, a couple had a child and 5 years later they want to have a child. So probably they can have copper T put in their body, as in the female's body. In that case, spacing would be there. So remember, two things are widely accepted. One is oral pill, and the second thing that you see is IUDs. Both of them are widely accepted. Do not get confused. So IUDs are ideal contraceptive for the females who want to delay pregnancy or space children. Correct. IUDs are most widely accepted methods of contraception in India. Correct. Both the statements 1 and 2 are correct. Okay, A is the answer for this question. Perfect, Sagar. Perfect, Mansa. What about the other children out there just listening to me? Okay, participate. Now over here, oral contraceptive pills mainly contain what? The oral contraceptive pills, which are also widely accepted, mainly they contain what? Remember, they contain progesterone and estrogen combination or progesterone alone. Remember, these hormones are really important for normal working of the body. But when you inject this, or when you give your body this particular hormone in more amount, what happens? The normal thing is not being balanced now. Everything gets unbalanced, which is why there are higher chances that the implantation would not take place, ovulation does not take place. Okay, and also, my dear children, even the endometrium is formed, it will be very thin. So there are few things that you need to remember: thin endometrium, no ovulation, no implantation also, because of these hormones. So these hormones can be taken in the form of pills, it can be taken in the form of implants where you put it under the skin, okay, which continuously release these hormones, and it can be also used as emergency contraceptives. Remember, emergency contraceptives have to be taken within 72 hours of sexual intercourse. Okay, 72 hours, do not forget it. Hello Rudra. Okay, now over here, oral contraceptive contains what? The answer for this question is B, progesterone and estrogen. It's not anything else. Okay, KCT sessions, ma'am, I want about KCT. We will start in the next week. Okay, after this unit test is finishing off, we will start it off again. We have certain things already being done. You can watch the videos if at all you want. Children, all of you watching this video, do you have your biology examination tomorrow? Do you have your biology examination tomorrow? I hope you have. Okay, now select the incorrect statement regarding vasectomy. Vasectomy means what? You're cutting off the vas deferens. Vas deferens is what? A duct that carries sperm in the case of males. Please remember, you have to remember all the ducts. Okay, remember all the ducts, the accessory ducts and the glands. It's very important in the case of male's reproductive system. Same goes with the female reproductive system also. So over here, you tell me. Okay, Muskan is saying yes, ma'am. Sagar is saying no, chemistry is there. Okay, I get it. So incorrect statement about vasectomy, my dear children, is that if you cut the vas deferens, that does not mean the sperm is not being formed. Sperm is still being formed in the testis. From the testis, the sperms come to the vas deferens also, but they degenerate over there because your vas deferens is being cut and tied. Okay, so please remember, the incorrect statement is that no sperm is there in the seminal fluid. That is correct. There won't be any sperm in the seminal fluid. The semen will be still released, but without sperms. That is correct. It's a surgical method of contraception. That is also correct. Vas differentia is cut and tied. That is also correct. No spermatogenesis. This is wrong. Spermatogenesis happens. Okay, spermatogenesis happens. So you should know that. So this is a wrong statement. No spermatogenesis. Okay. Muskan has physics and biology. Okay, wishing you all the best for the physics as well as biology. Now, question number 8. Children, this thing you can answer if you heard me correctly in the previous question. Okay, which of the following does not give the defined action of a contraceptive? Hormonal method prevents or retards the entry of sperm, prevents ovulation and fertilization. That is correct. Hormonal method. So hormones, whenever you give, that will do all this thing. It prevents ovulation, prevents fertilization, prevents or retards the entry of sperm also. How? Because the cervical mucus gets very thick so that sperm cannot enter. So that is correct. Vasectomy prevents spermatogenesis. Do you think vasectomy prevents spermatogenesis? No. So this is the one that does not give the defined action. So this is wrong. Okay. Barrier method prevents fertilization. Does barrier method prevent fertilization? Yes. Both the sperm and the egg cannot meet. So yes, it does. Now, intrauterine devices increase phagocytosis of sperm, suppress the sperm motility and the fertilizing capacity of the sperm. Yes, that is also correct. IUDs, okay. IUDs such as CUT, copper T, they help in phagocytosis of the sperm. They help in suppressing the sperm motility and also the fertilizing capacity of the sperm. CUT. Okay, if we talk about lipase loop, which is non-medicated IUDs, they help in phagocytosis of the sperm. They help in killing of the sperms. So please remember it. So all the intrauterine devices have different functions. I have discussed about this in the complete revision video, theoretical video. Go watch it. Okay, so your answer for this question would be B. Now, question number 9. Which of the following are not completely curable? Hepatitis B, chlamydiasis, HIV infection. All the options are given over here, and I gave you a clue in my sessions when I taught you this particular chapter. Okay, which of them are not completely curable? All of them. Sagar, if at all you remember, can you tell me? You are a, what do you call, consistent student out there. Which of them are not completely curable? All of them, they start from H. Okay, so it is hepatitis B. Over here, genital herpes as a H, and also HIV infection. 3 of them are the diseases which are not completely curable. Whatever you do, they are STDs, but they are not completely curable. Okay. Muskan, okay, I think that's previous videos questions answer you're saying. Okay, anyways, so here the answer is D. Now coming ahead with the next question, the specialized procedure to form an embryo in the laboratory is known as. In the laboratory, you're making the embryo. What do you call this process as? Gamete intrafallopian transfer, intracytoplasmic sperm injection, artificial insemination, wait, intrauterine insemination. The specialized procedure to form the form the embryo inside the, okay, there is some problem with this question. Specialized procedure to form the embryo in the laboratory is known as IVF, in vitro fertilization. None of these options can fit in this. Okay, it is known as in vitro fertilization. Okay, under that, if at all you had ZIFT, zygote intrafallopian transfer or things like that, it would have been correct. But over here there is no option, so I'll just move on to the next question. Okay, over here, out of the options given below, choose the correct stage for transfer to the fallopian tube for the successful IVF results. Out of the options given below, choose the correct stage for transfer to the fallopian tube for successful IVF results. My dear children, what do you do for successful IVF results? If your embryo is up to 8 blastomeres, you can put it in the fallopian tube. Okay, if it is beyond 8 blastomeres, you will put it in the uterus. This is known as intrauterine transfer. If you're putting in the fallopian tube, this is known as ZIFT, zygote intrafallopian transfer. It can be either the zygote or the embryo up to 8 blastomere stage. Okay, so your answer would be A. Fine. I think with that we have completed all the questions out there. My dear children, please make sure you are going through all the methods of ART, that is assisted reproductive technologies. Okay, that is ICSI, artificial insemination. If you have low sperm count, it is intrauterine insemination that you do. So make sure you are going through the key differences. As I told you, I have made a separate video. I'll be putting down in the link in the comment section. You can watch it. And if at all you think that this video was helpful, do not forget to click on the like button. Do not forget to subscribe to the channel. And also, if you have doubts still persisting, put it down in the comment section. I'll take it on priority. Wishing you all the best. Give your best tomorrow. Okay? Bye-bye. Take care. Sagar, I am not a good person to give tips for English. I think that if at all there was an English lecturer over here, I would have definitely tried my best to ask them. But yeah. So, bye-bye. Take care.